Hello, everyone. Thank you for joining our webinar today, Produce Testing. Your sample is presumptive. Now what? This webinar is hosted by Joelle Mosso, Associate Chief Scientific Officer for the Eurofins Produce segment. I'm Amy Knock, and I'll be moderating this webinar today. Before we begin, I just wanted to let you know more about how this webinar will run. The webinar is being recorded and the slides in the recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer viewer submitted questions. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions anytime throughout the webinar. So with that, I will pass it over to Joelle. Joelle, you can begin your presentation.
Thank you, Amy, and good morning or afternoon to everybody who's on the call. Thanks for joining our webinar. Like Amy said, my name is Joelle, and I serve our produce industry. So in anything respect to food safety related issues or quality issues, we're here to help. As Amy mentioned, the title of this presentation today is Produce Testing. Your sample is presumptive. Now what? And so that is likely get my slides why you joined. And so in that webinar invitation, we posed a challenge to everybody, and it's probably one that many of you experience in the produce industry. You walk the field or your pack shed and you pick some product, you drop it off the lab, and then you find out you have a pathogen presumptive. And probably the next question everyone thinks is, well, now what do I do with that, right? We all know in the produce industry is that we unfortunately don't have a kill step generally, so we don't have a way to get rid of pathogens after we harvest our products. So it's not that unrealistic that we found a pathogen presumptive. And then secondarily is that we know we grow our product in environments where pathogens live. It's kind of like the ocean. We know when we go in to swim that there may be sharks in the ocean and we might encounter them. So nothing all that shocking about finding a presumptive for a pathogen, but the question of what do we do with that is really more where our minds probably go after receiving that result. So that's what we're going to talk about today. What are we actually doing with testing in fresh produce? Knowing that we don't have a kill step and knowing that we are going to be encountering these sharks occasionally in our environment. The question that we're going to talk more about today really is, what are we trying to do with this testing? Why were we doing this test in the first place? And that leads us to, how did we get here? And the realities is that a test result is just the visible outcome of many decisions that we made far before we ever picked that sample and dropped it off at the lab, and certainly before we received the result. A test result really is just making something visible that we generally can't see. So we have these forks in the road, these paths of choices where we go one way or the other way. And intentionally or unintentionally, we make a lot of decisions of what our result may mean by how we take the sample, how we actually define our sample, what is it that we're actually looking for, and ultimately what tests we end up using. And some of these things may be things we don't even think about, right, on our day-to-day life experience. We know our customer requires a test, we go pick something, we go drop it off. So even unintentionally, we're defining these when we submit a sample. And it's important to know what they mean so that when you do get the result, you have a way to interpret what that result may mean in respect to your supply chain. So we'll start first with the first of those little bullet points. Test plan design. We're going to talk about surveillance testing, lot acceptance testing, and investigatory testing. And these are probably the last two, the lot acceptance testing and investigatory testing, are probably the ones that most people are familiar with, even though surveillance testing is probably my favorite to talk about, especially in respect to products that don't have kill steps. So we'll go through in kind of brevity a little bit about each of these and then see how they all tie in together. Surveillance testing basically determines normal occurrence of the target. So the word, prevalence, how often it might be there. So if you're thinking about E. coli in your field, let's say you have a 1% prevalence. So out of 100 samples, you would expect to find it 1% of the time. Another easy way to think about prevalence is to think about weather testing. So if you watch the news in the morning and afternoon, somebody talks to you about, well, we're 10 degrees above average. The only reason we can know that is because we have an understanding of the trends and seasonality with temperature. And that's also true in pathogen testing, or with any kind of testing, to be honest, is that there's a prevalence, and that prevalence may change based on weather, locale, what you're growing, basically micro ecosystems. The important thing about surveillance testing is that it provides a baseline of what we would expect to find on any given day. Because really what we're trying to do in scenarios where we don't have a kill step is that we want to understand when our risk goes up. Well, if you know you have a prevalence of 1% of E. coli in your fields, and you're finding it at 5 to 10%, something is obviously shifted. And that's the value of surveillance testing. It allows you to see something in more granularity. It can also, very importantly, be used to dictate appropriate lot acceptance sampling plans. And all of these are going to be based off of statistics. And so we'll go more into lot acceptance sampling plans next. So this is probably what most people are familiar with in the food industry, is that we take a sample, we get a result, hopefully negative, we file it away, and we accept the lot to go into commerce. Or if we get that pathogen presumptive or confirmed pathogen, is that we reject the lot because we assume that it is not safe to be consumed. It is assuming that whatever sample we took is representative of the rest of the lot. So if you think about an acre of lettuce, it's assuming that whatever your one sample was, however you took it, would be reflective of everything else that might be in that field. It is also assuming the prevalence is high enough to be detected. And so that's that connection to surveillance testing. That prevalence information is really important. If it's not very high, you'd have to take a lot of samples to be sure that you would accept or reject a lot appropriately. We'll go more into that statistics or those statistics later, but it's an important thing to remember. The other thing to think about with lot acceptance testing that we often don't, simply because it's, we're moving on our day-to-day life, is that cumulative data from lot acceptance testing can actually show trends in prevalence. If you're doing sampling correctly for lot acceptance testing, it will actually populate the same distribution of your prevalence over time. And it may take a long time, depending on how many samples you take. But in theory, that's what you should be doing, just at a slower rate. Investigatory testing is probably the next most common that people are familiar with, and that may be more commonly called root cause testing. So you have an issue or something you want to explore, and you decide that you're going to do additional testing to better understand that scenario. So perhaps you got your pathogen presumptive, and then you want to go sample the beds next to the bed that you sampled the product from, or the canal water, or the irrigation, or the drainage ditch next to you. That would be root cause testing. It can also be done if you're starting to question, well, maybe my lot acceptance testing, maybe my sample is not reflective of my true risk, because maybe my surveillance testing or what I know about from the industry, that surveillance is higher than what I'm seeing with my lot acceptance testing. So it may be something that you want to identify a better sample. We'll go more into that in a couple more slides, but the important thing to remember is that a test result only means something if your sample is reflective of the rest of the group. A very common type of investigatory testing that we talk about, especially in respect to food manufacturing plants, is vectoring or explorational testing. So vectoring, if you think about like Listeria monitoring in a drain, you often hear, well, take your vectoring sample. What does that mean? And often it means going concentric circles out, or directional sampling to see, is it moving into or out of certain spaces? Is it getting out of the drain into other parts of your facility? So all very important tools in our toolkit to manage for food safety. So now that we kind of have an idea about sample design, we're going to talk about the sample itself. And I know this seems like starting at the very basic, but it's actually a pretty critical thing to think about, is that a sample is only going to be reflective of what that sample represents. So it's very important that you pick a sample reflective of what you're trying to assess. If you might imagine, let's think about cattle for a second, is that if you're doing beef testing and you want to know if a steak is safe, you're probably not going to swab the hide of the cow, right? You're actually going to look at the meat itself. And you're probably more likely to find a pathogen on the skin or the hide of the cow. So the thing that's important when you're taking a sample is to design it such that you're successful in getting the result you want to see. So in the case of this romaine, maybe you want to test the tops of the leaves. Maybe that's what your sampler goes and pulls, and then that's the reflective of your sample. But the question would be for you, is that reflective of what your consumer may eat? Or maybe it's lower on the romaine. Maybe it's a lower on the different ribs or the outer leaves that you may strip off. Or maybe it's at the bottom. The important thing to remember in produce testing, much like what I kind of led with in the beginning of this discussion, is that we don't have a way to control our whole ecosystem when it comes to produce testing. And the same is true in, if you think about the micro ecosystem that is on this plant, it will be different at different layers and levels on the romaine head. On the top, you're going to see high UV exposure and relatively low water. On the bottom, you may be more subject to cross-contamination from the soil and collection of water in the crevices of the leaves. So if you think about that and look at your samples, that you may have a very different ecosystem at these different levels within the head. And all of that is stuff that should be considered when you're taking your sample. If you always sample in a certain way and it's not where the organism is that you're looking for, you will never have a reflective rate of risk. Could be your whole head, right? Some people sample the whole head of romaine, and then it comes to the question of how does your laboratory process it? Does the whole head go into the process? Do they chop it up, etc.? All of these things have some pretty big impacts on how and what your results may be from a sampling event. So now that we have our kind of design, so are we doing surveillance testing? Are we doing lot acceptance testing? And we've defined what our sample is and how we're going to collect it. We also now need to think about the statistics of how it's distributed in the field. And so here we have this lettuce field, and we have these different bacterial cells, and they are uniformly spread across on this field. So if we walk this in a Z or W pattern and we're pulling our product, we have a good likelihood of picking one of the samples, and it would be reflective of the rest of the product in the field. So if we found one, it would be good because we can see on this picture at least that there are other positives that should be removed from the marketplace. But what if they look like this? In this case, it doesn't matter if I have the best sample. So if I'm taking the whole head of romaine or whatever, is that realities are if I'm only taking a sample, I'm not likely to find it given this low prevalence, which we'll talk a little bit more about later. What about this? We have a higher prevalence. There's more positives on this field, but they're not equally distributed. And so the important thing to remember is that a lot of our testing methods and sampling methods assume that we have this uniform homogeneous prevalence. So that if we take one sample, it truly is reflective of something else. Well, so when you get that sample result that says you're presumptive and you have a homogeneous or a representative sample, you can say, yes, I'm glad I found it. I don't know if there's anything, I would believe there's other things in the field. But the realities are is that sometimes we might find this, where we found this one sporadic event, maybe it's just inherently there, and we have no idea whether or not it's throughout the field or if the rest of that field was actually clean. So these are all things that are assumed, often not even really thought about when we're doing testing. Which gets us to some statistics, and I know that's no one's favorite subject, or very few people's favorite subject, so I'll try to be brief and then give you a more easy to understand example. N equals 60 is something in the produce industry we talk a lot about. So N equals 60 samples that we're going to look at. Why do we talk about that? Because it gives us a 95% confidence rate. I know I've looked recently in certain testing documents, and that's what it effectively describes about N equals 60, which is true if you have a 5% prevalence rate. So if everyone just thinks back, for a second of how many times you find positives on your product. If it's not 5 out of 100, then you're not at 95% confidence rate with an N equals 60. So let's assume here that you're at 2%. So the top numbers here. So 2% defective, meaning 2% of your samples have a pathogen. That would mean if you're doing N equals 60, that 30% of the time you're missing what you're looking for. So 30% of the time you're releasing product into the market that may have a pathogen, if you're at 2% prevalence rate. So earlier on in this presentation, one of the things I mentioned about surveillance testing and why it was really important to know prevalence is that when you know prevalence, you can actually define what is an appropriate lot acceptance plan. If you don't know prevalence, it becomes much more difficult to have a statistically relevant lot acceptance plan. And why does that matter? Well, lot acceptance, if you're going to accept or reject a lot, depends on how much confidence you have in that test result. So knowing something more like this can help you define a better program. If you are at 5% contamination rate, then you have only a 5% chance of accepting a product that has a pathogen that you did not detect, which is pretty good, and which is why a lot of people use the N equals 60 level. But again, it's assuming that you're at a high rate of pathogen positives at 5%. If you're lower than that, you can see how that impact changes. And this is from the ICMSF, which is the most commonly cited sampling document, the book on sampling for microbiology in foods. So the important thing is to find your role and understand what you're really trying to do and have a better understanding about what your averages and your prevalence rates are, because it may change throughout the year and you may need to dictate and change your sampling plan appropriately as well. To drive it home in a little bit of an easier, less statistical manner, I like to use the Where's Waldo. So for those of you who aren't familiar, these are books that have these pictures that the whole goal of it is to figure out where Waldo is in the picture. And obviously it's pretty difficult. There's a lot of different other people that are there. And Waldo's the gentleman here we see on the side. So you might imagine if there's only one, so one Waldo in this picture, that if I randomly or even 60 times put my finger down, I'm probably not going to land on Waldo. If there are 100 of Waldo on this picture, I have a much higher chance of figuring it out. So it is extremely dependent on knowing what the likelihood of finding something is in your field about how many samples you really need to take. And then you have to think about how you actually take that sample and what is the true sample. Is it the whole head of romaine? Is it the bottom? Is it the tops of the leaves? How can I design a program to be the most successful to find Waldo? So what's our purpose? Surveillance, lot acceptance, investigatory, all of these will dictate what that result means when you receive it. What's your sample? Make sure it's reflective of your product. We need a representative sample that will make sure that you don't make decisions off of something that is truly not representative of your whole field. And if you have a whole acre being represented, or even 5 acres, this is a pretty critical piece that you want to make sure you're receiving the best sample that is most reflective of all the other product that's in the field. Even a perfect sampling plan, so how many samples you take and how you define what kind of surveillance program you're doing or sampling design program you're doing. It doesn't matter if your sample isn't representative. If I'm actually testing the soil and not the product, then that becomes a non-representative sample. The last bit that we'll talk about now is how do you test? And this is a pretty critical one as well. And all of these decisions, some of us don't even think about when we drop off a sample at the lab. So every test has trade-offs. And the important thing is, how do we choose what's right for us in the right situation? So first we'll start with the concept of tools. This is a bathroom scale. I think we're all pretty familiar, and it's technically a tool. So a scale will tell us weight. What it won't do is gain or lose weight for us, unfortunately. So we get on and off the scale, it will tell us what we're at, but it won't change what our reality is. Some scales are more accurate than others. So if I'm trying to lose 5 pounds and this is a 10-pound increment scale, I'm not going to be very successful at knowing if I've hit my goal. So the important thing is to know the limitations of the tool you're on. If it's a 10-pound increment and I'm trying to lose 5 pounds, I will only know I'm successful every so often. The other thing to think about, we all know this trick, right, is that we weigh different amounts during the day. And it might make us feel good to know that we weigh at a certain time a certain lower amount, right? But the reality is that we do weigh a heavier amount later on in the day. And this is true in testing as well. If you test in a situation where you get the feedback you want, a negative result so that you can release product, is that it doesn't change the fact that at a different time of the day or in a different way to select the sample is that you might actually be positive. So testing, again, won't change what realities are. It just shows you what you're at, just like a bathroom scale. So we need to take into all these considerations when we're choosing the right tool for what we're trying to assess. In produce testing, because of the speed to market that we all have to meet, right? So there's a short shelf life and it's hard to coordinate harvest with our testing getting done. We're often looking for the fastest method that we can find in order to find whatever we're looking for. And so in our perspectives in microbiology, that's one colony forming unit in a 25 to 375 gram sample. So there are different variations of the sample weight itself. So one cell effectively in that amount. Most current rapid methods require us to get to a growth step to detect that. And we'll go through this in a second. But basically, you need to get to 1,000 to 10,000 cells. And there's a lot of things that can impact growth. Injury, if the cell is not in a great state, so it's injured, it's got a lot of competition. So if you think back to that romaine head, is that there's often 10 to the 7th, 10 to the 8th CFUs of other bacteria on those leaves. So that's a lot of competition. Many other cells that are going to be competing for the same resources. The limits of biology. So I often talk about this is that for anyone who's ever had a bad haircut, how do you make your hair grow faster? If you can, to be honest, and if you can, please let me know, because that would be something I would love to know. But the truth is you can only synthesize new cells so quickly. And so that's what really becomes the limitations of our rapid testing, is how fast can we grow one cell to many cells so that we can detect them. We have some amazing molecular technologies to detect these pathogens, but we still are dependent on growing them up in an enrichment media to effectively just make that needle that we see in the haystack even bigger. To kind of drive this home more, I'll give you an idea about with Listeria. This is from a few of our senior scientists here, Dr. Doug Marshall, David Legan, and Danny Marco. But if you have one Listerial cell in a sample size, let's just say it's a 375 gram sample, because everybody wants bigger samples, and you look at the average weight for a Listeria cell, you're actually at 4 times 10 to the negative 20. There's a whole lot of zeros there. You can see in grams in a 375 gram sample. But what does that actually mean? Well, if this was a 6'6", so a 6'6" person, they'd have to travel 100,000 body lengths or 120 miles to get to the other side of the 375 gram sample. And just for reference, the state of Delaware is 96 miles long. So that's a pretty big fact. So if you can kind of just like think about that for a second, about how small one bacterial cell is in a very large sample. So this 6'6" person is having to go 120 miles to get to the other side of the sample. That's why this is a daunting feat. So science must deliver methods that can detect as few as one cell in a sample as large as 375 grams and accurately detect it so that you don't miss it, because that's what you're trying to do with your sampling program, right? The other thing is to talk about enrichment microbiome profiles. So I'll describe quickly here. So we talked about rapid methods being needed in produce testing. And that's because we just honestly don't have much of the luxury of time. Unfortunately, time is what allows us to grow up our target so that we can find it in that enrichment. So that one cell in 375 grams, say. So what's happening in these enrichments when we are growing them up? So I talked about how on the outside of romaine leaves or other lettuce products or leafy greens or apples or whatever produce item, these are not sterile environments. In fact, they're very dynamic ecosystems. There are a lot of other bacteria in there. And this is an enrichment microbiome actually of ice cream, but you can imagine the same thing for any kind of produce item. Is that all of the different colors represent different species of organisms and how they change over the time of an enrichment. So here you see at the 0 point on the leftmost side of your screen, where and what is in there. And the dark blue on the very bottom is what our target is, Listeria. And then you enrich it and you can see over time, is it in this solution that has a lot of nutrients? So you can see how the dynamics change of the population. And over time, especially around time point 28, 32, and 36, you watch that your target actually gets at a much higher level, but that it is always there even in the beginning. So as we start to push for more rapid methods, what we're actually changing isn't the detection method, the PCR, the molecular method that we're looking at. We're changing our likelihood of finding it, so more statistics. Because the sooner we look, the less time we've given our target organism to grow and the more competition it may have in its enrichment. And this is certainly what we see here. So around time point 32 hours, we pretty much have Listeria, and we're very likely to find it when we pull out an aliquot to test it. But earlier on, we won't. And this is why time is such a critical measure in any rapid method that you might use, is that understanding how your target changes in reflection of competitive microflora is an important thing to understand and something to talk about with your lab. The other issue in produce testing is because of the large volume of samples, some people choose to use wet cooling. So what do I mean by wet cooling? So those enrichments, we have our samples, we add our nutritive liquid so that we can grow up our bacteria, and then we incubate them at higher temperatures to select for them. Well, some people remove an aliquot of those enrichments and mix them together to then do detection. So here you see 5 spinach samples, and they're each separately enriched. And we're going to take out a small portion, 10 microliters, so very, very small portion, and we mix them together to do 1 PCR or ELISA reaction. And in this case, we see there's no pathogen, so we don't detect it, we move on, and all is good with the world. But what happens when you have one of these samples that may be positive? And what happens when that positive is actually at the lower limit of detection? So if you think back to that enrichment dynamics with all the different colors of different organisms, is that it's possible early on, especially at the shorter enrichments, that we won't be far past limit of detection, in this case, 1000 CFU. So we have one sample, 1000 CFU. We then take an aliquot of each of these and we mix them together. And suddenly we only have 200 CFU in that mixed enrichment, which means we actually are shipping a contaminated lot. We just don't see it because we've changed the limit of detection. This is particularly common, or this is particularly something to think about when you're looking at rapid methods that keep it shortening and shortening the enrichment methods. It's really important to understand what might be happening. What about if it's higher? So let's say we're at 10,000 CFU. Well, the limit of detection is 1000 CFU. So at 2000 CFU, which is what we would be diluting it to, we may find it, but we may not because it's subject to sampling statistics, right? So the question is, how confident are we in whatever result we're getting? Is it negative? Are we confident that it's negative? Is it positive? Are we confident that it's positive? Remember that with any test, there are limitations and we need to understand them in our discovery and decision-making process. Every test is just a tool and they're rarely perfect. The key thing is to know the upper and lower limit and to make sure that they align with whatever our sampling design program is for. When we receive that presumptive, it's important to know what it means. And so you need to recognize the trade-off and understand that every test, there's no test on the market that doesn't have limits and assumptions. The important thing is to understand what they are. So that gets us to what do we think about when we're going to be designing our testing program. Now, in the produce industry, we often talk about testing for the pathogens, and largely that's because we have more rapid methods for them. And then also is that it's pretty common, like I talked about the 10 to the 7th, 10 to the 8th CFU, per gram on romaine. Well, there's a lot of other indicator organisms on there that may not mean anything if I were to do testing for it. The key is to figure out what are we trying to understand. Are we designing a surveillance program? Are we designing a lot acceptance program? Or are we doing investigatory testing? And depending on our desire for what we want to understand, our targets may be different and may change. Or there may be more creative ways to track and trend it. One of the things with pathogens that's helpful is that they do have rapid methods, and that if negative, you feel confident or at least comfortable releasing product, albeit maybe less so after listening to the statistics of N equals 60. But if you get a negative Listeria or Salmonella or your big 7 for pathogenic E. coli, you feel good that the product, at least that you tested, is not there and you ship. but it doesn't help you understand as trends as they may change. So some people also look at other indicators in the case of generic E. coli or coliform or Enterobacteriaceae. So as you can see on this chart, you have different opportunities to look for different things, some of which may be better indicators for other organisms. And in addition, there's other pathogenic risk assays that you can do to be more comprehensive in what you're looking at if you're trying to do more investigatory or understand surveillance. So these are things to think about when you're designing a program. It's pretty easy to go through your kind of day-to-day existence for just meeting whatever your customer spec is. But the realities are is that if you receive that result and you're not really sure what you're measuring, maybe think about measuring something else. So something to talk to your labs about and your contacts and your experts to see if there may be something that you could do to drive more value in your testing program. So that leads back to your presumptive positive. What does it mean? Well, if you're doing a PCR method, which is what most people in produce are doing, what it effectively means is you're detecting DNA of that organism. And I always kind of like to equate that to, you know, the crime scene investigation unit, CSI kind of shows, is that what that means is you found evidence that something is there. What it doesn't necessarily mean is that it's living. And so there's additional methods you can do to understand if it's living. And some of which are, you know, DNA processing. So you can do some processing to see if it's just exogenous DNA. So DNA that you find in the environment, but is not bound within a living cell. And then what we would call cultural confirmation. So if you've heard confirmation, that then takes days to happen. That's where we're growing out the organism to make sure that we didn't just detect the DNA signal that it's there. Important thing to remember about PCR testing is that DNA is a signal, it's not a sentence. It doesn't necessarily mean that the organism is living. So these are discussions or things to think about as you interpret your results. It's also then important once you're holding that positive sample, you know, result is really what does it mean in respect to your program? And to be honest, you don't always know the answer to that question in the day that you're receiving the result. But the important thing is to use it in a way that drives the most value for you. So are you going to be doing surveillance testing or adding it to your surveillance cumulative data set to better understand trends and tracking? Is it lot acceptance? Do you need to put product on hold or do further investigatory testing because of your positive? I would encourage is that if you don't know these answers as to whether or not this is what would be expected to be found or not, is that you do some investigatory testing to better understand your environment, to know if you think back to those earlier slides where we had the grid of the lettuce field, is it one cell when you find it or is it many? And doing some investigation testing can help you get that. Honestly, the produce industry is really difficult. The environment that you're growing in is very dynamic. And so therefore it may be undefined, but what it is a data point that in cumulative can mean a lot as a data set. And so that actually becomes where your value is in retrospect after the testing. What are the limitations to your tool? And this isn't just in respect to your positive, right? So you do need to think about is that generally as a lab, we get questions when people get positive, but people rarely question the negative. But truthfully, there's limitations on both sides and understanding the sensitivity and the specificity. So is your positive really positive and is your negative truly negative? And what are the limitations of how you're running your method? Are all very important discussions to have with your laboratory when you're picking your different methods. And there's lots of different methods that come out into the market all the time. This is a highly technological industry where we're seeing improvements day over day with new approaches and new ways to do things. So talk to your labs, talk to technology companies and kit manufacturing companies to understand the limitations and really choose appropriately the best tool for what you're trying to understand. Use your data as a data set and not as a data point. This is a big one that you've heard me say many times throughout, is that even a lot acceptance program over time should build what your actual true prevalence is. If you see discrepancies like outbreaks and recalls that don't match what you should expect to see in your lot acceptance program, think about how you're taking your sample. Think about what your sampling plan is designed to find. Step back from your plan. Define success and how to measure it. make sure you select the tool that measures what matters. That's a really important one. Develop a plan for when a positive is found. There's the things that you do right after finding that positive that are obviously reactionary to the positive. But don't forget to use your data set in retrospect and understand if you could start to identify trends. It's definitely the case in my past histories where we've seen different issues in different ranches or different lots or with different seasons. Once you understand it better, you can define a better sampling plan. It may be that you don't always do the same sampling plan because your risks change, your prevalence changes. Drive for value and not activity. More isn't always more, and certain times of the year, maybe it's not. Certain times of the season or certain scenarios, it may not mean better if you do more sampling. The key is to focus on value. Where do you get the most and best information? And never be afraid to change approaches. That's what your data set is going to tell you how to do. So it's really important, even though it's very challenging, to go back and to look at your data as a cumulative, but there could be a lot of really good information in there that helps you design a better program. And most importantly, helps you understand what does your result mean when you get a positive. Ultimately, all of this means that you're designing purpose for your testing. And what leads us back to our prompt that led to this webinar. You know, what do we do? You're walking the field, you pick some product, you drop it off. What does it mean when you find out that you get that sample presumptive? The key is we're here to help. So reach out to Eurofins. We can help you understand your data set and think about your data set in a way that drives value. Testing doesn't change what your risk is, but it does allow you to see it so that you can change your behaviors. And really the biggest piece of information that I'm trying to drive home with this presentation is that a sample result can mean a lot of different things. The key is to use it in the way that's most valuable. And if you need help with that, you know where to find us. So I would like to thank you for the presentation today and open it up for questions, Amy.
Thank you, Joelle, for your presentation. We did have a few questions come in, and again, I invite, if anyone has anything else, feel free to type that into the Q&A section. So the first question, Joelle, is I currently do product testing, but I do not know what it means when I find a positive since I do not know my normal prevalence. What do I do with that information?
Yeah, and I mean, I'd love to say there's an easy answer to that, and that's partially what makes product testing really challenging in any category that doesn't have a kill step. So the important thing is, and on that day, you may not know. The important thing is to really start to rest upon your data set and start to look at trends and your cumulative patterns that you may see. The realities are that if you expect or feel like it's normal to get one positive every so often, and you feel like that's okay, and you suddenly are getting a ton of them, that's when you can start to really use your data set to say something different is happening here. And really, as we extrapolate forward, thinking about testing and what does it mean in the future, is it gets to that point of being predictive, where you can understand in any given day or scenario where your risk may be higher. And that's extremely valuable because then you can change your testing program to be more reflective of that risk. So unfortunately, no easy answer on that given day, but the truth is that we all should have a good understanding, especially after years and years of testing, of what a prevalence rate might be, whether that be environmental or on product. And if we're seeing discrepancies between in the industry, these outbreaks and recalls and what we're seeing in our own field, we may want to think about our sample design, our sample plan, and the true definition of what our sample is. Maybe there's something we could modify that could allow us to find it better or more efficiently. So those would be the things that I would say in the short term, is to start to look back at your histories, understand your trends, and then see if they match what your expectations are.
That sounds great. Thank you. And this question again about prevalence is how to find prevalence initially. Test the entire batch?
Right. So I mean, a lot of this, especially in respect to produce, can change from locale by season and maybe crop rotation as well. You know, what your outside environment looks like. But yes, effectively is that if the best way to get to prevalence is to have a good understanding of the scenario you're in. If you give kind of a comparison to environmental monitoring within a facility, a lot of these what we call a swabathon event, where we may use indicators that would not be exactly the pathogen, but we'd have an idea of how often we should find a certain level of something. And so you go into the plant, you do, you know, hundreds of swabs or whatever, and then you have a better idea of, well, this is my normal. Maybe you do that a few times to have some sort of understanding. I've heard, you know, some people in the industry looking at different generic indicators just to see if they understand different areas that have different issues. So the first thing to do if you're trying to get an idea of prevalence, besides talk to industry and research groups, of which, you know, Center for Produce Safety and many, you know, published journal articles can help elucidate, is to just do some of your own testing off of indicators, or do more testing on an exploratory field that you wouldn't be releasing to product, and get an idea of what you may expect to find or not find. If you're not finding too much, think about indicators. If you think back to that Where's Waldo kind of picture, is that it's a lot easier if you're looking for people who may look like Waldo, so Waldo's entourage, as opposed to just Waldo. It gets easier to find it, and that's the concept of indicator testing. The broader the group, the easier to find it if there's more of them. So there's different approaches, and we're happy to help, you know, design custom approaches if need be. So feel free to reach out.
Wonderful, thank you. And then I think that there are a few more questions here. So this question says, my lab uses the terms presumptive and a confirmed pathogen. Aren't they the same?
Right, so with most of our rapid methods that we have in the industry is that they are often DNA based, which is that crime scene investigation. So, you know, everybody, if you think about it in that respect, is that if I leave a hair at a crime scene and that is what implicates me being there, my DNA is there, I'm not physically standing there, my hair was. And that's the same thing with pathogen testing, is that it is possible that what we're detecting is DNA from a cell that was there. And there's still value in knowing that something was there at some point. The important thing, though, from a food safety implication, is that that thing is no longer living, which is where culture confirmation comes in. So while PCR is extremely sensitive, over 99% sensitive, what it isn't reflective of is if an organism is living. And so culture confirmation is where we actually take the enriched sample, the one that we did PCR analysis on it, and plate it out and try to culture it, grow it out onto plates. More of the traditional biochemical analyses of microorganisms. Why the industry doesn't just start with that? Well, quite frankly, those take many, many days to get even some sort of result. And so when your lab uses presumptive, it means we found DNA or indications that something's there. And culture confirmation is more the old gold standard, traditional of growing something out. Is it living?
Okay, great, thank you. And then I think that there's one more question that I saw come in. How do I know if my sample is representative?
Well, this is kind of the same, there's the same scenario that we're talking about with surveillance testing, with sampling testing. So one of the slides that I had early on in this presentation was a rooming head, right? So if we think about our samples, especially produce, they're outside generally, is that there's an ecosystem within that plant. So it's part of the large, a larger ranch, and that ranch is part of a larger area, and then that area is part of effectively the world. And at every level of that, there's a different ecosystem, and that's true on your sample as well. So you could do some investigatory testing, and I know some scientists have actually done some of these studies, but to understand where your greatest likelihood of finding something is, to understand how to take your sample. So what is the piece of the product that is actually your sample? And then where does that plug into? How do I know if it's representative enough? Well, that comes into play with all of the other factors that we talked about. How many other samples are you taking at the same time? If you were to test them all, are they consistent or different? Again, a lot of this comes from using the data set that you may already have, as well as investigatory testing to define and better understand what truly is your sample. If you're always taking the tops of the leaves, that's not reflective of what's lower in your product. If you find that there's more microorganisms or the specific ones that you're looking for that are lower on your plant or in a different area of where you're not sampling, that's not a very representative sample. You can do thousands of those samples, millions even, and not find what you're looking for. Where one distinct shift if you do a study to figure out where am I most likely to be successful finding it. It could actually mean an enormous amount, which could actually cut down on your testing. If you're just better at finding it, and that you understand when you find it, what it means, then you could be more successful designing a better program.
Great, thank you. And I've just been monitoring and I don't see any other questions coming in. So I think at this point, we will go ahead and wrap up our Q&A session. And I just want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available within 3 business days. And I want to thank you again, Joelle, for sharing your presentation and expertise. And again, thank you everyone for attending and have a great day.